Aujourd'hui, nous avons le plaisir de recevoir Hiram Castillo Michel. Merci à toi. Hiram est chercheur au Synchrotron ESRF à Grenoble. Il a réalisé son master de chimie et sa thèse en sciences environnementales à l'université d'El Paso, au Texas. Après un an de résidence au Synchrotron de Berkeley, en Californie, Hiram a rejoint l'ESRF à Grenoble en 2011. Il travaille depuis sur la ligne de lumière ID21. Hiram s'intéresse à l'application et au développement d'outils synchrotron pour l'étude de la distribution des éléments dans des échantillons complexes, environnementaux et biologiques. Et grâce à ces outils, Hiram a été un des premiers chercheurs à étudier le comportement des nanoparticules en interaction avec les plantes. Il continue à s'intéresser au devenir et au transport des éléments potentiellement toxiques pour les plantes. Et depuis 2019, Hiram est en charge du programme de mise à niveau de la ligne de faisceau ID21 pour développer un nouveau microscope à rayons X pour la nano-analyse qui est en service depuis février 2024. Je laisse la parole à Hiram.
Merci. Ça fonctionne? Oui. C'est bon. Merci beaucoup. Merci beaucoup pour l'invitation. Je vais passer en anglais, si ça ne vous dérange pas, pour ma présentation. Par contre, questions et tout ça, après, on peut discuter en français, c'est juste pour être plus à l'aise pendant la présentation. So, thank you for the invitation. It's for me a pleasure to be here. I have for a long time collaborated with Camila Wu and now with Astrid, and so I hope you find this presentation interesting and maybe you come to ESRF as users to do some interesting project. So I will start by answering quickly the question, what is the synchrotron light? So synchrotron is a facility in which you have an accelerator of electrons. So electrons is the particle that is accelerated to almost the speed of light. And then it's injected into a storage ring in which you have different magnetic devices which are forcing or guiding the electrons to go in this circular path. And when these electrons are deflected, you have emission of light. And so this light is very collimated. It's collimated and is very intense. So, for example, in comparison to an X-ray tube, which is more or less what you would find in common laboratory instruments that are using X-rays, you have 12 orders of magnitude until you reach the new state synchrotron facilities in the world. And there is a big course to go into the diffraction limit storage ring. So a lot of the facilities in the world are upgrading so that they can provide even more light. So these are photons per 0.1% bandwidth in a very directed direction, very small space. So a lot of photons. And what can you do with all these photons? Also, you emit light from infrared to X-rays, so you can select. So it's all this electromagnetic spectrum light. This is very advantageous. So then after you generate, so in here you can see this is the storage ring, and you have the different deflectors, so different magnets, and then you have the beam lines. And so the beam lines are very large infrastructure facilities in which you will fine-tune this or select the lights or the X-rays, in the case you are only using X-rays, that you will use for your experiment. So in here I'm showing you have some insertion devices and bending magnets, and the difference is that the insertion device or an undulator makes the electron to undulate, to deflect several times, whereas a bending magnet is just one strong magnetic device that just makes the electron to detour once. And so you will see that beam lines at ID21, they are called either ID, so insertion device beam lines, or bending magnets, BM, which are coming off bending magnets. So that depends on the source that you're using for the photons you will use in the machine. So also, how many synchrotrons are in the world? So I don't know exact number. I think it will be about 40. So you can find more details in this website. But here you can see all the locations in the world. You can see Europe it's kind of packed, or there is a lot of sources. But particularity of ESRF is that there is only 3 sources in the world that have the same size, so the electrons have the same or close to the same energy, so 6 to 7 giga electron volt. So one is in Chicago in APS, and the other one is in Japan. And so this leads me to what is the particularity of the European Synchrotron Radiation Facility. So first of all, it's the only facility in the world that does not belong to only one country. It is hosted in France, but it is funded by many countries. So you have 17 participating countries, of which some of them are member states. So France is one of them, as a main contributor, followed by Germany, Italy, etc. And then you have associate countries. And so this goes beyond Europe and is not necessarily related to EU. And so you have, for example, Israel and South Africa, which are a bit outside the European territory. And any other country could at some point approach the ESRF and become a member or an associate country. And so you have 42 public beam lines. And in 2015 or 16, a project was done in order to make this facility an improvement of what we call the extremely brilliant source. So it's just to get more of the electrons packed into a smaller beam. And so in here, what we see is really the X-ray emitted by the previous machine, which was this large, very horizontal source, to this one, which is a bit more symmetric. And so for ID21, which is where I work, this means that we went from something which was 2.8, 0.28 millimeter to something that now is squarish. And this means like no light is lost. All the light is packed into even a smaller. And so this gives you even more photon density, and you can do more interesting experiments, higher resolution, better detection limits, etc. And so this is the team. Some ex-members. The team right now is composed by Marine Cot, which is being responsible working in cultural heritage marine. We have an engineer, leading operation manager, Maria Salome, technician. These are 2 former postdocs, which are now beamline scientists in other synchrotrons in Europe. And Clement is our new postdoc. And me, I am a scientist, mainly working in environmental science. And we have our position open. So if you know anyone who is interested, then your picture could be in there. And the domain we are looking is something from environmental or biology or science, something about, you will see the applications I have chosen, is someone to come and work in this research domain. And so back to ESRF and all the beamlines, you see you have so the photons are emitted tangentially to the acceleration of the electrons. So we don't work with electrons, just to be clear. It's light. The X-rays is photons. The electrons are the source, but we never make our samples to interact with the electrons. And so all along you have all these beam lines. And so I work at one beam line, so I cannot give you details about all the techniques and all the experiments that are taking place all along and at the same time of the synchrotron facility. But I could give you just quickly some of the beam lines that can help you to get either lateral resolution covering from millimeter all the way down to nano to 25 nanometers, I think is the record on ID16A. You have a microfocus, nanofocus like ID21, and then this is energy range. So you see the beam lines kind of overlap. So there's no beam line in the world that has the same capabilities. So when you want to do an experiment, you have to make sure that this experiment fits into the beam line, or on the other hand, you should look for the beam line that can solve the questions you're asking. And so in here, in terms of chemistry, how you go from X-ray diffraction to X-ray absorption spectroscopy, or X-ray absorption fine structure, or even high resolution X-ray emission, etc. So these are complementarities in terms of like the chemical information, and these are complementarities in terms of like the lateral resolution. So it depends on the question. You need to choose wisely or appropriately, accordingly. So ID21, so what can you do with ID21? So ID21, we are an X-ray microspectroscopy beamline. So we use 2 techniques, X-ray fluorescence imaging. So we detect the photons emitted by the sample, and we do X-ray absorption spectroscopy. So if you look at, in terms of spatial resolution, we go to now 200 nanometer. So in terms of spatial resolution, you can see subcellular details. In terms of detection limits, we are at the level of a femtogram. So, okay, in ppm, this is a complicated conversion, but let's say imagine like femtograms, so very, very small amounts of material or elements we can detect with high resolution. And in terms of specificity, when you combine this with X-ray absorption spectroscopy, or more the part that we call SAINS, then you get details about the element of interest, the chemistry. This is like oxidation state, what is the geometry of this molecule, and what are the neighboring atoms. And so in terms of the energy range that we cover, we go from 2 to 11 kiloelectron volts, and I will show you in the periodic table what this means. We operate in vacuum, so the samples need to be vacuum compatible, which should be done, they have to be vacuum compatible. And we operate in cryogenic environment, and we have 2 end stations, and I will give you with the details the differences between the old microscope, already 14 years old, but still quite good performance, and the new one, which we just begin to do experiments in February. So this is back in. So the 2 microscopes are in line. So the old one is first. Then after you can just like put optics out and open the valve, and then you go into the nano end station. So we could, let's say, switch relatively easily from one to the other, and we can choose according to your sample, the sample that you want to analyze. And so in terms of resolution, over here we are more at the level of, let's say, 500 nanometer, and in here we can go down to 120 is what we have recorded, but let's say in practice, in theory, we should go to 100 nanometer. So 100 nanometer, but also this one is, because we go more in the nano resolution, is more stable for long-term analysis, and we have more detectors, so the detection limit is also better, and you can go faster, because we don't want to provide, let's say, only like 10 micron for you to do at 200 nanometer, but we want to maintain the possibility to mount relatively large samples, and I will give you details about this. And so these are the 2 microscopes that we offer, and all quite still like kind of all-terrain vehicle, and this one, which is like a new Ferrari, but you have to be careful what you want to do in this one, because it's really for nano analysis. And so this is the way the nano analysis station looks like in CAD, in SolidWorks. So in here you have the focusing optics very close to the sample. From here to here you have only 30 millimeters, so everything is super packed, and it was very difficult to come up with this design. Then in here you have all these detectors, and this is ×2, so there's another detector here from the side. And then in here you mount your sample, and this is for you to visualize it, and you get a resolution of 0.8 microns, so it's a very good visible microscope. And then you have X-ray present detectors, and detectors that help to detect the incoming beam and also the transmitted signal. And you have a cryo stage, which is always in place, so we can cool down the sample. Obviously, it takes time for everything to become stable, so we can't do this like room temperature to cryo in one experimental session, but everything is in place. And so this is the way it looks more in reality. Here you have the KB optics, video microscope, your sample mounted, and this is evidently this is a room temperature mounting what we have here. The samples, your samples will go in this. And how does this look? It looks like this. You have, let's say, about 16 millimeters in the horizontal direction, and about 8 millimeters for you to put samples. So whatever you can fit in this 2-dimensional space, and in 3D, we can give you a bit more space. But for the high resolution, you never want your samples to be too thick. But if you're analyzing soils, minerals, etc., usually they are embedded in some resin and polish. So even if they are a few millimeters thick, we can still mount them. And so we are just developing different sample holders. So if you have an application that would benefit from this, the analysis at our end station, you can always contact us in advance, and we will try to produce something where you can you can mount your sample. And so for the cryo setup, we have this. Everything needs to be in metal. So we have aluminum, which is a good thermal conductor. And you have this area, but this is again, we are working on how to make it larger so that you can also in cryo analysis benefit from all this analysis space. Okay, so just to tell you a bit about the detectors, why the new machine is more sensitive than the other, because we have 2 detectors, and in here, this little thing here is what we get out of our old microscope. And these red curves is what we get now in the new one. So it's it's an order of 5 or 6 better detection, which you can use to go for things which are more diluted, or you can go faster. So in principle, with this machine, should be able to run with integration times on each one of the pixels of your image to the order of 10 milliseconds, whereas right now in this one we are going at 0.1 second, so it's 10 times faster. And so this is in terms of chemistry or elements, what we can do. So we go from 2 kiloelectron mole to 11 kiloelectron mole is the high limit. So basically all the elements which have color we can detect, but all the elements that have color are underlined, we can do X-ray absorption spectroscopy on them. So you can see, like, you have from phosphorus all the way to gallium, all transition elements here, and then we have some of the heavy and the rare earths. Of course, we have to consider detection limits, etc., interactions, what is the matrix, etc. But at least you can see, if you are interested, say, in cadmium, you can from here see that we could do analysis on cadmium, for example. So this is a very, rather than this energy with time, this you can convert into something, but with the periodic table, you can very quickly see what we can do. And so how this works, so we have basically, we scan a sample, and in each one of the pixels, we will record an X-ray fluorescence spectrum. And then after, you can get images which are giving you the distribution of the different elements. And then after, we go in the spots, and then we collect the X-ray absorption curves, which, as you can see, can tell you details about the chemistry from iron 0, iron 3, etc. So in here, I will try to explain a bit much better. So you send, so after this is the beam line, your sample is what is scanned, the beam is fixed, you have a detector. So after you send X-ray, you excite, you remove an electron, and with it decays, it produces fluorescence. And then this fluorescence, it's processed by a pulse processor, and then you get an histogram with the intensities. So this is the intensity of the photon and how much of it. So this peak is proportional to the concentration or the presence, the amount of the element that you have in the sample. And so you can create a region of interest in given peaks for the detector, and each one of these peaks represents an element. So in here you have the superposition of titanium, calcium, and potassium, and this is the vascularity of a root. And so after that, you can display these only in like false colors. So the red is the most intense spots of titanium, and you can bring the beam into this location and then register the X-ray absorption spectrum. And this X-ray absorption spectrum, because X-rays do not need things to be crystalline, they can be crystalline, liquid, gas. It's any state of matter, they will interact, they will produce a spectrum. So you have sensitivity even for things which are not crystalline, don't have any like crystalline order, but also between even though it's the same, for example, in here titanium oxide, you have anatase and rutile, and you can distinguish these two. So even if just the crystalline order of the same elements changes a little bit, you have the possibility to distinguish this with X-ray absorption spectroscopy. So it's a really interesting tool for chemical speciation. And so I would like to talk a bit about this cryogenic sample environment, because it's very important for environmental samples, for biological samples. Usually, like if you could do analysis in native state, then that would be the best. But at the moment you want to go high resolution, it's a bit forbidden. You stop at some point. And so what you can do is to do cryofixation and cryosectioning. And for that, you need to embed your sample in some sort of resin, called OCT, and then plunge freeze it. And then after you have to section. So at ID21 we have a cryomicrotome, and we provide users with support to do this sort of preparation at the beam line. And this is the cryo stage of the old microscope, the SXM. And so in here you see you have all these copper parts. So you have liquid nitrogen in anywhere somewhere here, and you're cooling down, passively cooling down your sample, which you introduce in this copper block, or aluminum. We can use aluminum as well. And so this is a bit how it looks. In here I have a leaf, then the leaf goes in the OCT, then you plunge freeze. This is isopentan in there, then it freezes quickly, and then after you go in the microtome and you get a slice, after the slice, you mount it on the holder, and this is a leaf slice into the holder that it's ready to be transmitted, to be transferred into the microscope, and then we can do the analysis on this. So this is what we call frozen hydrated state analysis, and so this is the way it looks in the new machine, in the Nano SXM. Everything is in place. You still have some liquid nitrogen circulating, but the sample is passively cooled. We also have what we call a sample hotel, or it's just an extra space where you can put the sample while it waits for the analysis. This is quite advantageous that we don't have in the previous microscope. So now you have one in the analysis chamber. So this is all the analysis chamber, but one in the cold finger, in the place where you are actually doing analysis, and the other one is waiting to be transferred. And this should optimize your sample transfer. And in here again, we have some dimensions in there, but we could develop different holders depending on the type of samples. So this is a work in progress. This is the way it looks in reality. And so also in order to improve this sample transfer, because for now in the old microscope, in the SXM, what we do is we come with the sample in a little dewar with nitrogen, and we put a glove in it, and we grab it back with the hand and transfer it quickly. And we try to do this as fast as possible so that the sample does not get defrosted, and it goes pretty well. But in here we're trying to do better, so we bought this Leica shuttle and this cryo manipulator. So in here you have a copper block. This is what goes into the analysis chamber. This is the way it looks in reality, and all this is transferred. In here you have vacuum, you have liquid nitrogen. So now we don't touch with our hands, never the sample, which is also good for us because you can get your skin can be damaged by the very cold temperatures. So we have to be careful. And so, okay, what is the scientific domains at ID21? So I have chosen some examples from the field where I feel more comfortable and where I work more often. But you see, cultural heritage takes 18% of the proposals, environment, earth science, life science, medicine. So it's really between like cultural heritage and environmental research and life science that takes pretty much all the beam time, all the shifts that we provide at the beam time. So again, the biological samples are a main criteria main criteria for our decision making when we are optimizing our instruments, because we want to be as friendly as possible for this kind of samples. So we're not so much like a material science beamline, if you want. And so, okay, now go to some of the examples. So say like overall tracking elements in the environment. We know some of them are essential, other are non-essential. So you might be looking at it from the point of view like how you can make it cultivars more good, more enriched with certain nutrients, or how we can get rid of certain pollutions. And for this, you want to analyze from global to micro to nano scale. And so you can begin to do like soil sampling at the level of a continent, of a country, or just some terrain. And then from there, you can go to the high resolution as to say, okay, where is this element located in the plant root or in a trichome, this little hair of a leaf? And this like goes towards the more and more high resolution analysis. And so what we actually do at ID21, we try to solve the questions: where is this element of interest located, and what is the chemical species that you have in there? Because both questions are very important to determine how they move in the environment and whether they are toxic or more beneficial. And so one first example I will talk is related to, again, this kind of sustainable agriculture. Is related to the nitrogen cycle. We don't analyze nitrogen at the beamline, but we could have a look at, in collaboration with a group from Spain, Manuel González Guerrero Lab, we look at the nitrogen fixation by the root nodules. So this is a leguminous plant with the Rhizobia bacteria. It creates this new organ, which is a nodule, which has a very interesting morphology in terms of zones of activity. In here you have really the place in zone 3 where you have the fixation of nitrogen. You can see there you have lots of bacteria infecting the cells and establishing a symbiosome. And basically what you have is nitrogenase getting nitrogen from air and converting it into ammonia. And this is very dependent on iron. So what we look, it's at the distribution of iron. So this is a schematic. This is how it looks with histology. And this is how it looks when you do histology, but with X-rays. So you can see the cells, you can see this pinkish hue here, this color is iron colocalized with potassium. And you can see that the epidermis is mainly defined by the calcium distribution. So you have iron, potassium, and calcium. And so in here, with more detail, you can see the cells which are infected with rhizolia. And so what we do after is we select, so for example, in here you have meristematic zone, you have mainly like hotspots of iron, and then all these little pink points is where we do SAINs. So we click using our user interface, you click in many points where you want to collect the SAINs. Same is in here, and then after we use multivariate statistics to determine that if the two groups are actually different in terms of chemistry. And then, as I said, these X-ray absorption spectroscopy curves, they can give you hints about oxidation state, but also distinguish between like oxygen ligands or sulfur. So in this case, iron sulfide clusters are very important in nitrogenase, so we can determine this. And so it's very interesting when you can combine this technique with normal molecular biology approaches, in which you can turn off certain genes. For example, in this case, this publication, it's a bit cut. This publication, which is looking at ferroportin, that is helping iron to go into the symbiosome. If you block it, in here you see this is the zone 3. This is where you have the infected cells and actively taking nitrogen. And you can see that the distribution is less. You have less infected cells, and it's just because you block the ferroportin transporter. So besides that, if you do SAINs in the two regions, it's not very observable, but here you can see the PCA plots, and the colors basically represent different groups. So the speciation is changing evidently because you have less iron in the symbiosome. So iron is also in a different state. So iron distribution changes, iron speciation changes when you manipulate some of the genes. And so when you can combine your classical molecular biology with the synchrotron imaging, then things become interesting in terms of information you collect. So again, this is the way the plant looks. These are nodules. We section the nodule, and this is really, the sample is mounted in the microscope. This is the way we see it. Then after we image, so this is the histology with X-rays, and then you can see different vascularities, the infected cells, the part with the cells that become senescent, and basically before EBS and with the old machine, we perform this map with a resolution of 3 micron, and we count in each pixel for 0.1 second. So in 3 hours, you get this image. But in the new machine, with the new detectors, we can give a resolution of 1 micron, so it's already an enhanced resolution. But because we go faster, we can get this in 2.7 hours, so more or less the same time, but this is 1 megapixel image. So now you have much more information, so higher resolution, but the same field of view. This is really part of the philosophy of the new design. Like, we didn't want to give you SL with 200 nanometers. We want to give you the entire structure, but with higher resolution, which is not so simple to do. You have to put all your effort into making the acquisition faster. And so the next topic is about also agriculture and food safety, and it's related to cadmium. Now there is no regulations from the EU about the limits of cadmium in dark chocolate and in milk chocolate, and because of this, 30 to 50% of the beans which are produced in South America that may not be commercialized in Europe unless they are blended, which is big socio-economical besides environmental issue, right? And these are not contaminated sites. These are just regions which have slightly above, not even above, just slightly higher concentration, but below or at the level of the background. So it's not contaminated site. This is very important to understand. This is not contaminated site. It's just, a little bit 1 ppm against half ppm, etc. And also the concentrations are low, really low, but still you get cadmium in the cacao. And basically what you see in the soils is what you see in the plants. So this kind of linear relationship is interesting to investigate. So at ID21, for many years, we have been working on hyperaccumulator plants. Like this is Arabidopsis alleri, and you can see here the distribution of cadmium in the cross-section of a leaf, and we can do speciation, and we see that there is some cadmium bound to thiols or to oxygen groups, etc. We also know that calcium oxalate plays an important role in hyperaccumulator plants, because when you have calcium oxalate grains, you have cadmium. And this was published in Amaranth, which is also a hyperaccumulator. So from hyperaccumulators, which is what we could do before, because we didn't have the detection limits that you need for working on things like cacao, but also we work on wheat. And so in wheat, you see accumulation of cadmium in this crease region. This is the full grain. And in here, we also know that you have oxygen and sulfur ligands. It's not cadmium phytate that we know. So let's say you can have like edible and also hyperaccumulators. And in here, we were thinking, actually, we can work on these kind of samples which are native, like really from non-polluted sites, but also non-hyperaccumulators. And right now, we are working in cacao, and we just published a paper from Esther Blomayer. It was a PhD thesis in which they went and collected from field cacao plants. So we analyzed the leaves, the branches, and the roots. And just to give you one of the main results, we know that in the branches, you also have these calcium oxalate crystals, as I show you in the previous image. So in here, you can see the crystals in the branch. And this is strontium instead of calcium, but they are together in these oxalate crystals, and the cadmium is there. And so basically, these calcium oxalate crystals are involved in this cadmium storage mechanism. And it's actually interesting because the cadmium stays in there, it doesn't go into the part that we eat. So it seems like this may be some sort of mechanism of passivating or just limiting the amount of cadmium that goes into the edible part. So it is interesting. Now there's a lot of work that needs to be done in order to understand this much better, but this is how we can contribute toward this understanding of how cadmium moves in cacao, and then so that we can come up with some ideas as to how this problem could be solved, and we could keep importing these cocoa products. And if you combine high resolution, now you can see small, but this is at 50 nanometers. So this we went in another beamline which has high resolution, and you can see also the cadmium and the nice shape of the calcium oxalate crystals. So that we are for sure that these are calcium oxalate crystals that are binding part of the cadmium that is in the cacao. And so now this is a project that I'm working with Sonia Rodriguez and a group from Portugal, and with Astrid Avelan from here, from Toulouse. And it's related to this nano-enabled agrochemicals. So in vineyards, you use copper sulfate, and unfortunately you put sort of heavy amounts of this, and then it can be washed when you are watering the plants, and this ends up in the soil and can cause problem. And if you put a nanoformulation, then these particles attach better to the surface. You don't wash them so easily, and they have the same effect of preventing the mildew, the fungal infection. And so this is one of the topics. I don't have results in this. We were working on that. I will present something similar, which is localization of micronutrient in the leaf. And the chemical transformation, but looking at zinc oxide, which is a paper that was very recently published, like 2 weeks ago, which is about the comparison of zinc oxide with zinc oxide and the phosphate shell. And so basically, the long story short is that when you have this phosphate, you have higher mobility of zinc, and it transfers better in the plant. So it's good in order to deliver this important nutrient zinc. And in here you have the X-ray fluorescence images from exposures of 2 hours. So the particles are deposited in this. This is leaf cross-section. also cryofix in the way I showed you in the previous video, versus the phosphate. You can see that the zinc oxide nanoparticles, they are accumulated in the area where they're deposited, and then they go to the bottom. And in here, there's much less. And a similar thing happens where you're in 1 week, you have a lot of zinc into the epidermal tissue layers, as opposed to when you have the phosphate. And so the question is, you put zinc oxide nanoparticles in the surface of the leaf, where does zinc go, and is it still zinc oxide? And this is what we can answer, because we could have the distribution of zinc, but we can also probe the speciation of zinc. So in this case, at 2 hours, I'm showing you just something we're very proud, is the fact that we can go into a thing which is 200 nanometer. This pixel, this red pixel is 200 nanometer. And then we can go and in 1 minute, we can collect this spectrum. And the red one is the one that comes from the sample, and the blue one is the original zinc oxide nanoparticle. So we could do spectroscopy on nanomaterials. I mean, the level of 200 nanometer, which is our best resolution, which is not so trivial. It's actually quite difficult to do this. So this is something we can provide at ID21. But okay, so we found some at 2 hours, but when you do at 1 week, in fact, you don't see much zinc oxide. In here we have references, fumarate and phytate, and this is the zinc oxide. And these are all spots which were collected in the tissue, and none of these resemble zinc oxide. So there's no more zinc oxide. It has been transformed. Apparently, it goes first into some sort of phytate speciation, and then it goes towards fumarate. So first you have phosphorus, and then you have carboxylate. And so we can answer this, like where is it going in tissue, and what is the chemical speciation? So this is important. Of course, in the paper you have much more detail with other techniques, because that's important to combine with other techniques. It's always important. And so another example also related to nanoparticles, but this is like in soil. So this is other kind of type of samples that we can analyze. This group is from Australia. And so in Australia, they are looking at cobalt. They want to understand the natural cycle of cobalt. And so there's different locations where samples were collected. So these soil samples, then they are embedded in a resin and polished. And then we do maps. And then after we found cobalt in the maps, then we do spectroscopy. And so we want to understand what happens from the parent material, which is our sulfides, and then it gets oxidized, and you can have cobalt +2, cobalt +3. And the more active is the site, the more you will have like interchanging between the species, if you have input from bacteria, if you have plants, etc. So just to briefly show the result, first of all, what we found is that cobalt is colocalized with manganese, which was the first interesting thing to see. And then in terms of the speciation, these are different sites, and which one which is the two which are more contrasting is this quinsadip site, in which you have, this is the location of cobalt +2, and then this pink one is cobalt +3, and you have lots of in between. So all over the site, I mean, in the sample, you have like cobalt scattering, like it can be +2, it can be +3, and so far we have not found any correlation to what is happening, whether this is bacteria or what is really the cause of this. Whereas there's other places like this copperado site, in which everything is very homogeneous, copper +3, but in all sites, it's always with manganese. So it's cobalt manganese oxide, and we know they're nanoparticles, because first, in some of them, we have these very small spots, but the resolution does not allow us to say what is the real size of this. But if you combine these with single particle ICP-MS, which is what is done in the paper, then you get distribution of this. And so we know they are nanoparticles, we know they are composed of cobalt and manganese, and we know that they are +2, +3, and it all depends from which side you are taking. And a bit more in-depth interpretation you can find in the paper. Which is like has been published like also 1 week ago. And so, oh my god, the time. I'm not sure. Maybe I'm going too fast. No, it's fine. Yeah. Okay. So this this is another. In here you have cryofix cells. So I wanted to show you like another. We saw plants. We saw tissue. We can do plant tissue or animal tissue or even human tissue if you have the corresponding authorization from safety. And so you can work on tissue, and we can do this at frozen hydrated state, but you can also think about bacteria or cells, plant cells, or in this case, these are HeLa cells, so this is just a human cell. And in here, they're looking at the uptake of cerium into the cells. So the cells are growing in a membrane. You have cerium in there in the fluid, and then the cells begin to uptake cerium. And so this, what I'm showing here, is an image in which the spectrum is obtained by doing several times a region of this map, for example, here, I don't know exactly where it is, but let's say this part. And then in every one image, you have an intensity, which you can then go and collect this pixel here and get a spectrum. So this is like 2D science. It takes time. That's why it's not done in a very large region, but in each one of the pixels, you have a same spectrum. And it's very interesting in order for you to see more clearly the differences in chemical information, but with really in a 2D mode. So in here you have only one energy, so I can tell you only where is cerium, whereas in here I can tell you where is cerium-3 and where is cerium-4. And what I give with cerium oxide nanoparticles is cerium-4, which has this specific double peak here, versus cerium-3, which has one white line only. This is what is called white line. And so you can distinguish cerium-3 from cerium-4. So you can distinguish whether these particles have been transformed inside the cells, and these are frozen hydrated, so they are as close as they can be to the native state. They are not alive, but they are frozen, so nothing is changing, nothing is evolving. And so in this case, we saw that there is internalization after 24 hours, and then in some cases, in little places where seem to be lysozymes, you can have some dissolution of the cerium oxide, but it gets precipitated quite quickly and forms again cerium oxide. And so this is the last example, which is a very interesting one. It's a combination of ID21 and ID16A. ID16A allows you to do this nice tomographic, is like absorption contrast. So it's the radiography, but it's done on another mode of contrast, which is called phase contrast. So it gives you three-dimensional structure, but also you can do 3D X-ray fluorescence. So in here you have the 3 dimension of a cell. This is a fibroblast from the skin, and you have nanowires, and the nanowires are internalized, and the comparison is between nanowires 30 nanometer and 90 nanometer. And in here you have the summary. If you have 30 nanometer nanowire, when it goes inside, it crumples, it gets encapsulated into a lysosome, a sort of lysosome structure, and then it gets dissolved with time and slowly. And so these nanowires are less toxic, because if you change their diameter to 90, then what happens is they basically go as a lancet. They go into the cell and they perforate all organelles, and the cell bursts and dies. And so with this, you can do, let's say, a safe by design approach, in which you can still use these nanomaterials into screens and whatever applications. But if you put them at 30 nanometer, they are more compatible with biology at the moment when you discard your cell phone or whatever. So this is a little bit the idea. And so we know how they go in the cell, We have the 3D structure, but also we know that when they are encapsulated in these lysozymes, they begin to change, because initially we put silver at 0, so this is metallic silver, and with time it begins to transform into some sulfide, which is a very common chemical process of degradation of iron in metallic. It becomes sulfidized. And so with this, my last slide will be just to pass the message that yes, the synchrotron techniques are very interesting, but of course you can't, as you could not only rely on one instrument that you would have at your institute, you could not rely only on the synchrotron. And so 1 year ago, we were working with Astrid and Camille into showing okay, how does it look, a sort of workflow for the characterization of nanomaterials in plants, in which you have some of the bulk methods, and many techniques which come from the laboratory, single particle ICP-MS, X-ray diffraction, and some of them come from synchrotron. And these are, for example, beamlines where you could do this sort of analysis. So it's just to show that we should think, I think that now in the way science works is by doing this sort of approach of multi-analytical workflows, and where you combine whatever you have in your laboratory, but you also combine with other laboratories. In this case, I propose you consider synchrotron techniques for your research. And if you really have an interest in this, then just send us an email or contact us, and we will help you to understand whether the synchrotron, which is really a service, a user service facility, can help you in your science. And so with this, I want to acknowledge on this side this long list of colleagues, because this nano SXM project is like really working with people from all the fields, and it has been so interesting. So nothing could have happened if you remove a single name from here. So I don't have time to mention all of them, but just to say that we really acknowledge all this, the work of all these people. And on the other side, of course, all the examples I've taken from collaborators and users. So in particular here, the Astrid is present, and we don't have examples of what we have done with Camille, but it's really a pleasure to work with all these collaborators, and I hope that you find this interesting, and maybe you can come to a beamline. And thanks for the invitation, and thanks for your attention. And finally, remember, we have a postdoc position open, so you can maybe join ID21. And that's all I have to say. If you have any questions, on peut passer en français si vous voulez.
Merci Hiram. Donc, s'il y a des questions, allons-y.
I have two questions. The first is the detection limits for the imaging, especially for the silicate minerals and probably glasses. And the second is what kind of standards you are using and how they should be prepared for mapping.
Okay. So glasses, there is a lot of work on glasses from the cultural heritage community. So glass, well, could be a very complex matrix. So you have, of course, a lot of silicium, right? But I don't know if you, for example, I have users, they come here and they are looking at the incorporation of copper into the glasses, etc. So detection limits for, if you are interested in silicium mapping, I guess any sample would be just more than enough. It's going to be actually sort of crashing our detector, so we would have to remove some of the intensity, which we can do, modulate it so that you could collect something. But the interesting part would be if you have a glass matrix which contains some other element, and more in particular, if you would have things like, for example, glazes in porcelains or whatever, where you have specific layers of other elements, where actually the 2D mapping could become interesting for your research. So in terms of detection limit, it depends on, I would have to know more in detail your system to tell you, but let's say, it's really ppm or sub ppm. And then for references, you mean for imaging or for the Xanes?
For imaging.
For imaging, for imaging, we just have to calibrate the beamlines if you are interested in some sort of quantitative approach, right? So we just put a membrane with elements on top, and then we determine how our detectors are, how sensitive they are, and we can calibrate them to some like quantity reference. That's only done when you're really wanting to do quantitative. So you have to also look in the details. If your sample, we can do quantitative analysis, because not all of them are so easy. Let's say in principle you could do with any sample, but you have to be careful with certain things. Let's say, I'm sorry to say, like it's very specific, so I would need more details to be able to tell you more precisely.
D'autres questions?
What is the pressure on the beamline in terms of submitted to accepted projects? Is it higher compared to other similar beamlines where you can do claims and exams? You cited in your last slide, or is it lower?
I would not know how is it in other beamlines, but I can tell you the success rate is, I think, is 1 out of 3. And at our peak, at some point before 2019, before the shutdown for this upgrade of the machine, we were at some point at records of 42, 43 proposals. And so we can get maybe 10 experiments out of 40. So there's 1 out of 4. But right now we are going faster for certain, because for other experiments, actually people are just bringing more samples because they were always lacking replication. And so at the end, we give the same amount of beam time, but you analyze more because statistically you need it. So I'm not going to say that our experiments are becoming shorter, but some of them are. And so this is opening space for maybe 1 or 2 more projects per semester. So I would say, let's say 1 out of 3. It's a good rule of thumb. But we are actually now putting a lot of advertisements, so maybe it will become a bit more difficult.
Thank you. I wanted to know whether you are measuring boron also, if you are going to look at the micronutrients in plants.
Boron is difficult. Everything which is below silicium, it's difficult because our detectors, well, first of all, we can't do SANES, so we could map, but our detectors have a beryllium window. And so I think we can do sodium and magnesium, and then below that, then it becomes really complicated because the beryllium window will just attenuate everything. And we used to have polymer detectors, but they were breaking very often, and we said this is not a good investment. Also because most of the time what we want is to do mapping and SANES, and for the very light elements we can't do SANES. So we said, okay, let's stay in our niche. There are beamlines where you could do that, which are the soft X-ray beamlines. So I think Lucia in Soleil can do this, and also TwinMic in Electra. I think you could look into those.
Thank you. Allez-y. Sylvie, en ligne, il n'y a pas de questions.
Je ne m'étais pas endormie, mais... Vas-y. Je change de sujet.
Okay. Also, you have shown a map with the calcium oxalate and with the strontium concentration. Why did you show the strontium and not the calcium? And are you able to map, for instance, the strontium of the calcium ratio in some plants?
You could look at the calcium strontium. The reason is when you do cadmium, when you are looking at cadmium, you have to excite below potassium. So you have cadmium L and then potassium, calcium. So you would have to do an excitation because we very fine select the excitation energy, right? So in this case, we were at 3.5. If you want to see calcium, you need to go to 4 kiloelectron. But then the problem is that this is a crystal of calcium. It will saturate. It will not saturate. It will just like emit so much signal that then our sensitivity on cadmium is not so good. So we fine-tune everything so that we can be very sensitive on cadmium, even though we don't get the direct image of calcium. But because in these crystals you have substitution of strontium by calcium, which is a process that happens quite often in this sort of biominerals. So then the strontium was our marker to find this. But in fact, you could do the ratio. Let's say maybe you need to forget a little bit about the detection of cadmium, and then you can just look at distribution of calcium and strontium, and you can establish ratios in the different grains and determine which one has more strontium than the other, and whether there is a reason for this. This can be done to work on both elements. We didn't want to, let's say, ruin our detection limit on cadmium. That's why we didn't map calcium. And in the high resolution map I told you, they are actually looking at one is at the K shell electron, so they go to high energy, like really high energy. So you have cadmium and calcium at the same time. But then I'm not sure what happens to strontium. I think you lose energy with the area. I don't know. But this is why I don't present calcium, because actually I was not exciting calcium. But we did the correspondence, we are pretty sure that these are calcium oxide.
Thanks, Hiram. So I had a question that is not so much on the science, but more on the disposition you are mentioning, because there are students and postdocs in the room. What would you say is that, what are the criteria that are required to be a good postdoc at ID21?
You like working on weekends now? What we are asking is someone that comes from the field of if you have done any sort of analysis or your interest is any of the topics that I show, and you have a bit of experience on that, then you have a good profile. So we're looking for someone from earth science, environment science, or biology, or the three. Someone that wants to work on these cryogenic samples is very important, because it's me who has more the most expertise in the beamline, and we need more people to work on this, especially for this part, tissue sectioning, etc. And then, other than that, the eligibility is like no more than 3 years after your thesis defense. And then, if you want to join the synchrotron, yeah, for sure, part of responsibility is to do your own research. We don't impose a topic. You propose a topic, and you should be kind of very well autonomous to develop your own network of collaborations, so that you have something scientifically, you need to produce some papers, good for your career. And the other part is like, you have the responsibility to offer support to the users. So this is what we call local contact. And so you have to see it's a different kind of position, in which you are not just there in your corner working on your science. You have to provide support, even if this is just once every 2 months, but you should like this part. It's very motivating, actually, if you really like this, to get involved with other groups. And we try to give local contacts to the postdocs from the field where they are coming from, and not to put you in something which is really, you don't understand what is happening. But sometimes even from this you learn. But yeah, so you have to be willing to combine your own research with providing support. This is something we really need someone that wants to work in a team like that. This is mandatory.
And I have a second question from an experimental standpoint, so both for this type of positions, but also for users, I guess, when we come. There are people here that do, for instance, mineral formation at freezing or low temperature. Is it possible to do some type of in situ type of measurement on your SXM with this hotel sample? I don't know if related age for a bit. Is it something we can consider doing?
If you want to do some sort of aging in the cold temperature.
Yeah.
Yeah. Yes, it could be. I mean, actually, this is even for the biological samples. You have this process competing a bit. You have to be fast enough, let's say, because otherwise, I think after 24 hours or maybe 2 days, you freeze dry the sample. So in minerals, I guess that it's cool and in vacuum, 10 to the -7, but you don't stop fully reactions. You slow them down significantly. So yeah, you could consider something like this. Like keep the sample in there for some hours. It is not going to bother us to do that. Other kind of in situ analysis are difficult. The end station is not planned to give you a lot of flexibility. What we can give you flexibility is in the kind of sample supports. So we can work together on a sample support that fits your sample rather than imposing. This is something we are proud of. Rather than imposing that you work until, we try to adapt. But this, sometimes if we don't have it, then you should consider this well in advance, that we will need to do some sort of development. But,
question? Non.
Bon, mais écoutez, on va s'arrêter là, alors, en remerciant encore Hiram.
Merci beaucoup.
Une bonne journée à tous.
You are currently the only person in this conference.